Everyone, and welcome to today's lecture. In this lecture, we're going to be talking about the psychology of forensic identification. So as you know, forensic evidence is used in many trials, and the process by which the evidence is discovered and brought to trial is through forensic science. So what is forensic science? Forensic science is the collection, analysis, and interpretation of physical evidence. And although it may seem straightforward, these words, collection, analysis, and interpretation, if you look at these three aspects, which one might psychology have a role in? That would be the interpretation aspect. So one thing that has come to the attention of psycholegal researchers in the last 15 years or so is that some of forensic science doesn't seem to be quite as sciencey as we once thought it to be. So one question asked by psychologists is whether or not biases can affect the interpretation of physical evidence at crime scenes. So let's take a look at first how forensic identification is done, and then where there might be some opportunities for error in this process, and also talk about some of the most common types of forensic evidence. So first of all, forensic identification. What is it? So with forensic identification, you are trying to link physical evidence to a specific individual. And this can take many forms of these. Forensic identification have been used in the courts over the years, sometimes only to abandon them when the accuracy has been found to be lacking in them. So for example, bullet matching evidence used to be widely used in court, but in 2004 it fell out of favor. But, by that time it had already been used in thousands of trials. And the basic premise for bullet matching evidence was first you analyze the composition of the bullet at the crime scene. And then, you find a suspect and you find that, he has a gun that has some bullets in it. And you look at the bullets that he has in the gun. And the idea was that you wanted to look at the chemical composition of those bullets. And so if the chemical composition of the bullet from the victim at the crime scene and the chemical composition of the bullet from the suspect, if those, if those chemical compositions were the same, it might suggest that the bullets came from the same box. So the idea was that bullets in the same box would be extremely similar in these trace elements, but would be very different from other types of bullets. But then, actually, in 2004, the National Research Council showed that bullets from different boxes could have almost identical trace evidence. And also that bullets in the same box could be completely widely different in this trace evidence and chemical composition. So that really, blew apart the whole bullet evidence. So this leaves us with an overarching question of, what are these methods and are they reliable? And that was kind of an example of a case in which we thought bullet evidence was reliable until research showed that it was not as reliable as we thought it might be. So before we get started, looking at specific evidence, I wanted to talk about some terms that are commonly used in forensic science. So the first one we have here is trace evidence. And this is a term you see very often in forensic science. And essentially what it is evidence that is left at the crime scene. So these are things like fingerprints or DNA. So this is what the suspect has left at the crime scene, and this is what we are using as the evidence, the trace evidence. Next we have source attribution. So the goal of this forensic examiner is to determine, whether that trace evidence, where it came from, or in other words, to determine its source. So first you'll have the trace evidence, and then you will have this notion of source attribution in terms of trying to determine where this trace evidence originated from. So, for example, if there was you could compare trace evidence of a fingerprint trace evidence to a suspect's fingerprint and make a determination about whether or not they match, trying to find the source of that trace evidence. Another term that you will see in the forensic science literature is the term inclusion, and this is sometimes also called a match. So essentially, a forensic examiner would say something like, the DNA at the crime scene is a match for the suspect. This is also known as inclusion. Sometimes forensic examiners will use even stronger words here, so something like individuation. So this means it's not just a match, but that in the process, they've also excluded all other possible sources. The only source that this could come from would be the suspect, no other sources. So in other words, the trace evidence is so matched to the suspect that it can only be the suspect that would match. There's no other person on earth that can match this trace evidence other than the suspect. Another term you might see is excluded. And excluded means that there is no match between the trace evidence and the suspect. And this doesn't necessarily mean that they aren't guilty. So for example, an exclusion might occur if there was, if the trace evidence was degraded for whatever reason. But if you do have good evidence that hasn't been degraded, then an exclusion really does mean that there is no match. And finally, we have inconclusive. And this basically happens when the evidence is either incomplete or contaminated or degraded in some form. So basically, if evidence gets contaminated while forensics examiners or others are trying to collect it, basically that means that it could lead to an inconclusive result, and that means that no decision can be made one way or the other. So a forensic examiner goes through this process of determining a match, and then after they do this, they have to communicate this information to the jury or the jurors in some way. So the scientists make this conclusion about the trace and the source, I have a match or I don't. And then they must communicate both the nature and the certainty to a judge or a jury. And that's typically done, there's typically 4 different ways to communicate forensic evidence. And the first we have is qualitative. And so qualitative means this is some type of just subjective statement saying things like, the match was weak, or the match was moderate, or the match was strong. One example of evidence that might use this type of qualitative statement is given about bite marks. So the forensic examiner really is only told to say things like, it was inconclusive, or it's improbable, or it's possible, or probable. And sometimes these words sound legitimate, but there's really no standards for deciding, which statement of those qualitative, subjective statements to use. So the degree of bite mark match is subjective. So that's really, that's why at most you can really only use a qualitative statement when trying to communicate this evidence to the judge or the jury. And next we have a simple match. For example, say there is a crime in which a perpetrator broke into a window and, tiny shards of glass are found in the perpetrator or the suspect's car, and those shards of glass are consistent with the glass from the broken window at the crime scene. So that is a match. The shards of glass match. But we really don't have certainty of a match, to be honest with you. So for example, it would be useful to know maybe how common that type of glass is. For example, if that type of glass was found in 90% of homes in America, if 90% of homes in America have that glass, then those shards of glass in that suspect's car mean a lot less versus maybe if this was some new type of glass that the window was made of and that was found in the suspect's car, maybe only 5% of homes have it, then this would be much more important. So when we're talking about simple matches, it's important to keep in mind, how important is it? Another type of communication, in order to how to communicate forensic evidence, is through a match plus statistic. And this basically takes the simple match, but it brings in some sense of certainty. So you're given a probability, a specific probability, that this match could be wrong. So a match plus statistic might say something along the lines of, there's a 1 in 1 million chance that this is an incorrect conclusion. So for example, we know that red hair is less common than black hair in our population. So finding red hair might mean more than finding black hair. So it's really about knowing something about how common this trace evidence or this piece of evidence or characteristic is. But unfortunately, we don't always know how common certain characteristics are in our population. And then the final type of communication is known as individuation. And with individuation, this means that this is the most complete match to the trace. So it requires scientists to be able to say, it's so detailed and so perfect that it could only have come from this one person. So this is the ultimate match. This is the most conclusive type of match. So now we're going to go on and move in to talk about some types of forensic trace evidence. So first we have DNA. And DNA is the most objective trace evidence that we have. So when I say objective, it means if we go back to that first page when we were talking about collection, analysis, and interpretation, DNA really covers the first two there, and then there's very little room for interpretation with DNA as well. So it's probably the most objective evidence that we have. And DNA was discovered in 1953, and it was first introduced into the court system in People versus Wesley in 1988. And it's not important to remember the particular case, but I do want you to kind of take note of the date there, because later on we're going to be looking at the Innocence Project, and look at people who have been wrongfully accused and then later exonerated with DNA evidence. And you'll see that many of them committed crimes before that 1988 cutoff. So many had DNA evidence collected, but it was not used in court. And since 1988, we have constructed an extremely complex database that is used by the FBI. And the most common database is the CODIS. And the FBI maintains this database. And if DNA is found at a crime scene, it is sent and it's put in this database and analyzed. So let's take a look at what they're doing here when they're profiling this DNA. So this graph right here is called an electropherogram. And what you can see is that it shows these different peaks. So see these peaks at these different locations. And these are peaks on specific locations of genes. So what they're doing is they're testing these heights in these peaks on several locations on these gene pairs to see if there's a match. And if there is a match, then the conclusion is pretty straightforward. But sometimes there is ambiguous results for DNA evidence, and this could be because of poor quality of trace evidence or contamination of the DNA evidence if it wasn't collected properly, and this may lead to inconclusive results. But the analysis done with DNA is actually done on a computer, and it uses these algorithms so that if there's a match and it's pretty unambiguous when you have, properly collected uncontaminated DNA, then there's really no room for interpretation there. So let's say that we have a match. How is this communicated to a juror? Because these calculations use massive databases, and they also take into account the frequency of each allele in the population, it allows us to make a match plus statistic or even an individuation communication. So this is usually almost always done with the probability statement, and this essentially is the estimated probability that the match could have occurred by chance alone. So what will happen is, for example, the odds of the DNA, the communicator may say something along the lines of, for example, the odds of the DNA from the crime scene not being from the defendant is 1 in 12 billion. And how they do that is really a simple probability estimate. This is something that you can do mathematically. But as you're going to see, the math explodes pretty quickly, and this is something that, a computer really needs to do. So if you think about, let's just think about two alleles here. What are the odds that there's a match? It's really a simple probability statement. A match or not match is kind of like a heads or tails here. So the probability is 0.5. So if two alleles matched, then you say, what's the probability that they could match even if this wasn't actually true? What's the probability that this match could have occurred by chance if this wasn't actually true? And with two alleles, that would be 0.5 times 0.5. So about a quarter of the time, you could get a match that wasn't actually a true match. But now, say we have four alleles. That would be 0.5 times 0.5 times 0.5 times 0.5. So the probability that all four match just by chance, is getting a lot lower there. And then you can see if you compare 8 alleles, it goes down even further. So again, you can see pretty quickly how these probabilities, would escalate so high to maybe this 1 in 12 billion, because the machines that are doing this are matching on many, many, many more than just 8 alleles. So that's really why you can get into these probabilities of 1 in 12 billion. So usually, as I mentioned, the forensic examiner will describe this match in terms of a probability, and these are known as random match probabilities. And this is presented to the judge or the jury, and as the name somewhat suggests, It's really just them communicating, what are the odds that this was just a random match. And this is presented to the judge or the jury when the case is tried. And this can be pretty convincing to the jury, you can imagine. This, 1 in 12 billion statement. So DNA, is probably some of our best forensic evidence, and it is well documented and well studied. Okay, so let's move on to a slightly different type of forensic evidence, one that is also well known, and that is fingerprint evidence. And this type of evidence has been with us for a very long time. In 1892, Sir Francis Galton published a book called Fingerprints, oddly enough, and this described the pattern of fingerprints. And Galton argued that every print is unique and that the prints do not change over the lifespan. And he also pioneered the procedure of matching fingerprints by counting points. And based on his work, he talked the British government into adopting fingerprints as a method of establishing identification in criminal trials. So you can see that, fingerprint evidence has been around for quite a long time. So let's look at, the basic pattern of fingerprint evidence here. So there are several patterns that are analyzed by these experts when they're examining them. So essentially what they're doing is they're analyzing the ridges, and there are 3 basic types of ridges. So first we have the loop. So with the loop you can see that the swirl comes in and the ridge enters from one side, so it enters from this side, and it goes up and it comes back out the same side. And this is in contrast to the arch. So you can see with the arch, it enters from one side, say the left side here, and it goes up and it forms the ridge, and then it goes out on the other side. It exits on the other side. So it comes in from the left, goes out to the right. Whereas with the loop, you can see here, comes into the middle and comes back out on the right here. And then with the whorl, you can see that the whorl here is really just this circular type of ridge. So those are the 3 basic patterns of fingerprints that experts look at. So how do these experts make an identification? Well, nowadays the fingerprint is fed into this computer with this large fingerprint databases, and then basically it flashes when there's a potential match. But the computer doesn't make exact matches. They usually spit out a couple of potential matches based on these algorithms. So then a human examiner has to examine these potential matches from what the computer has suggested and pick out the actual match from there. So as you can see right there, there's a potential place for a mistake or bias or any other factor that could lead to a mistake. So some errors really some human errors in identification really can cause issues and these false identifications. So you're gonna watch a short video this week on the Madrid bombing, and this is where there was a mismatch of fingerprint evidence which led to the arrest of somebody who did not commit the crime. Luckily the actual perpetrator was found, but certainly you can have these misidentifications or these false identifications with fingerprint evidence because of this human examiner who has to make this decision. So there is some potential for error there. So let's look at, what these experts really try to examine. This is a set of fingerprints in which an examiner has determined that there was a match. So first you can see, they kind of compared the ridge characteristics, and you can see that the characteristics that are noted to be very similar here. You can see on this picture with these numbers, these are the characteristics that they noted to be similar across these two fingerprints. So they look at, things like bifurcations, where the ridge is split is very important, and sometimes they look at crossovers, and you can see that this happens, at various points. They've determined that this happens at various points in between these two fingerprints. But how do they decide, yes, I have enough here that there actually is a match between these two fingerprints? How many ridges need to be similar for me to say, yep, that's a match, or nope, that's not a match? And this material is kind of qualitative. They do look at things like the print size and the shape and the orientation, along with the number of similarities that we see numbered here. And you can see here, right, in this specific case, there were said to be 12 similarities between these two fingerprints in terms of ridge characteristics. So 12 is, quite a few, so the examiner might decide, yes, there is a match here. But that decision is still pretty qualitative. However, many other countries do have not in the US, but other countries do have a specified number of ridge characteristics that have to be similar to say that there is a match. So some countries, these kind of range from, say, 7 matches to 16 matches. So, in some countries, there has to be 7 of these points, versus in others, you have to have 16 to say there's a match. So this is definitely a bit more quantitative. But the problem is, overall, with fingerprint evidence, is that we have this assumption that we've never really addressed, and that is, whether or not every fingerprint is actually absolutely different. And we also don't really know how common or rare each type of ridge characteristic truly is in the population. So if 90% of people have this exact marking, this type of ridge characteristic, then that makes it a lot less interesting than, say, we know that only 5% have this exact ridge characteristics. So, while fingerprint evidence is fairly well documented, there still can be definitely mistakes made and room for error there. And in fact, if you look at research on biases in fingerprint identification, it can actually be a little bit scary. There's not a lot of research on this, but with what little we do know, it's kind of scary because a lot of people put a lot of faith in or weight on fingerprint evidence, but the data doesn't really support this, and the science really leaves a lot of room for error there. So for example, there was one study that used untrained participants. So these were not fingerprint experts, they were just untrained participants. And what they did is they exposed them to some emotionally arousing stimuli. Such as gruesome, graphic crime scene photos, or they exposed some participants to non-emotionally arousing stimuli. And then they looked at these fingerprints, and they had to make match decisions. And they found that those participants that were exposed to the emotionally arousing stimuli before they had to make these decisions were more likely to say that the two prints that had been presented to them matched. So in other words, this suggests that basically if things are very emotional, then there seems to be this urge to find matches between fingerprints where none actually exists. Another study that was done used fingerprint experts. So these weren't just laypersons, these were actual fingerprint experts. And specifically, these experts were given 8 fingerprints that they had previously seen before, and they had made a determination. And half of those fingerprints had been declared to be a match previously, and half had been declared as non-matches. Additionally, half of them were easy to judge, and half of them were more difficult to judge. So they had some easy ones and some hard ones. And then, finally, half of them, they were given misleading information about a particular print. So saying something along the lines of, the suspect with this print has already confessed. And so, in other words, these were 8 fingerprints that they had before, and they needed to determine whether or not there were matches. So they simply asked them to look at the fingerprints and say whether or not there was a match. And the results were this. Only 1/3 of the examiners were perfectly consistent in their judgments. So, they said exactly what they had said before. So that means that 2/3 of the examiners, a majority, gave inconsistent results with these determinations. And the changes in judgment that did occur, they occurred most often in the difficult print condition, which makes sense, but then also when they were told the suspect confessed. So you can see here that knowing that the suspect had confessed biased their decision-making on this task. So we need a lot more information here in terms of fingerprint evidence, and we need a lot more information in order to understand the variables that are at play that might lead to these biases in identification. And lastly, I just wanted to quickly touch on a couple of techniques that are known to have either weak or unknown validity. So we just went over, some of the 2 most common types of forensic evidence. We had the DNA and the fingerprints, but there's also other kinds that are used, but we really just aren't sure how valid they are or if they are valid. And even if we do think that they have some validity, the data suggests that they're very weak techniques. So for example, we have the analysis of bullet striations. These are still used in court today, but there's no standards. There's no standards for how large the striations have to be or how many types of striations need to match in order for there to say it's a match. So really, when we're using these types of, this type of analysis in a courtroom, we're really just putting our faith in the examiner. Another type of technique might be bite mark matching, or examining different tools that have been found at the crime scene. And, these are just data shows that these are weak techniques. So for example, trying to determine whether this knife at the crime scene, or this knife that I have in my kitchen is the same knife that was used at the crime scene. These kinds of decisions and matches are being made in court today, but there's really not a lot of evidence that those conclusions are not heavily influenced by that interpretation aspect that we've been discussing. So now that that was just a short little preview of forensic evidence, but I hope it gives you a better understanding of how forensic evidence is used and can be used, and the fact that there really is a lot of interpretation and bias, even in something that can seem to be pretty clear-cut and dry.